You're doing biology at home, you're just doing it remotely via Canvas. It's not ideal, but it's what we have. If you're at home, you can always access these PDFs, and you can then mark up the PDF like we showed you. I know it's not what you might prefer, so a Google Doc is sometimes another option that you can mark up the Google Doc, which sometimes you might not prefer. But if you don't have access to a printer to print those things at home, then that's what you got. Now again, the expectation is that if you're not here, that you are staying up to date. Everything's posted in Canvas. We spend a lot of time in Canvas making a detailed agenda list of what we have with links to each and everything we do, in addition to posting videos. When you return though, just so I thought we had said, but all of our paper handouts are underneath on the top shelf right by the fire extinguisher. So in the event that you need a handout from one of those, that's where you can access the paper copies. Okay? Now, I'm going to be honest that unless I get an email from you while you're on quarantine or whatever, my expectation is that you are staying up to date, that you are learning along with us. Now, if you're really sick, email us. Let us know that. Like, hey, I've been really sick. I've gotten behind. I'm doing my best to get caught up. We understand. But if you're on quarantine and you come back the day of a test, I'm going to hand you that test because my expectation is that you are learning along the way. Without communication, I am assuming you are staying up to date because we're posting this stuff in Canvas. If that's not the case, you need to tell us that you're not and why you're not. Maybe it's an internet issue. Okay, well then that's another thing too. Maybe it's you're really sick. Right, absolutely, communicate. Okay, paper copies are always available over there, so when you do return. But the expectation is that you do and you get caught up on the stuff that you missed. Okay, so prokaryotes versus eukaryotes. This is on this packet on page 4. Okay, let's take a look at page 4. So on page 4, we have a chart. And I want to make sure we have the right information because it was kind of all over the place in terms of did we have it or did we not have the correct information. So I just want to go down those columns. Okay, let's talk about what things, based upon our pictures, because that's what we're doing, this is not prior knowledge, based upon the models that have been given, what cell organelles can we find in bacteria cells? And there are 3 types of bacteria cells, but they all have the same common organelles, I'm sorry. So, does a bacterial cell have a cell membrane? Yes or no?
Yeah.
Yes. Okay, so we can either write the words yes or no, which is what it says, or we could do like a check mark. It's kind of up to Mr. DeBoer what he's going to choose to do. He's going the Y versus N approach. Okay, so all bacteria cells have a cell membrane. Do bacteria cells, which are prokaryotes, have ribosomes? Yes or no?
Yes.
Yes, they do. Do bacteria cells have cytoplasm?
Yeah.
Yes, it shows that they have cytoplasm. Do they have a mitochondria?
No.
You should be making these corrections. Make sure your chart is right. This is a nice compare and contrast of the two. Venn diagram is something that you might see here in the near future of pro versus eukaryote, and this is a great kind of reference for that. Let's start with, we'll skip nucleolus. Let's talk about nucleus. Does a prokaryote or does a bacteria cell have a nucleus?
No.
No. Within the nucleus is a structure called a nucleolus, and it produces ribosomes. But if there's no nucleus, is there a nucleolus? No nucleolus. Okay? No nucleolus. Does a bacterial cell have DNA?
Yeah.
Yeah, and according to the model, it said it was free floating, which means it's floating around freely. It's not contained in anything, hence the lack of the nucleus. Cell wall. Did
Yeah.
all of them have a cell wall? Did all 3 pictures have this labeled? Yeah, it did. Okay, it had a cell wall labeled on all 3 of our pictures for bacteria cells. Is a bacteria cell a prokaryote or is it a eukaryote?
Prokaryote.
Okay, so we put a Y next to prokaryote. That's really it. Is it a prokaryote? Yes or no? And then, is it a eukaryote then? No. And that memory device that they used on the video, pro and no, pro and no, they kind of rhyme, no nucleus. Okay, another way to think about it. Okay, let's talk about animal cells. Remember, you are an animal and you are eukaryotic. Does an animal cell have a cell membrane? Yes or no?
Yes.
How about ribosomes?
Yes.
How about cytoplasm?
Yes.
How about someone besides Garrett? How about mitochondria?
Yes.
There we go. I like the choir kind of approach. Do they have a nucleolus?
Yes.
Yes. Nucleus?
Yes.
Yes. DNA?
Yes.
Yes. Cell wall?
No.
No. Okay. Are they prokaryotic?
No.
Nope. Are they eukaryotic?
Yes.
You betcha. All right. Plant cells. Cell membrane.
No.
Yes. Okay. So if we look really quick, right here, there's an outer cell wall, which is in dark, but then just inside the cell wall is the cell membrane. Okay. So plants do have a cell wall, but they also have a cell membrane. They have both. Ribosome.
Yeah.
No. All right. Cytoplasm.
Yes.
Yes. Mitochondria.
Yes.
Nucleolus.
Yes.
Nucleus.
Yes.
DNA.
Yes.
Cell wall.
Yes.
Yeah, they do have a cell wall. Prokaryotic?
No.
No. Eukaryotic?
Yes.
Yes. Okay, so now we're just kind of looking across the rows. Did we put yeses for everything? For cell membrane? Yeah, all cells, any type of cell has a cell membrane.
Yes, all the way down to DNA.
All cells, no matter what type, have a ribosome. All cells, no matter what type, have a cell cytoplasm. However, there was no mitochondria in a bacteria cell. There was no nucleus, nucleolus or nucleus in our bacteria cell. There was, yes, there was DNA in all. That's the genetic information that gets passed on from generation to generation. And not all, because animals don't, have a cell wall. Okay? Not all cells are prokaryotic, and not all cells are eukaryotic. Okay? Those are some kind of common mistakes. That wasn't, we were kind of all inconsistent with this. So I want to make sure we have the right information before we kind of move on to this. But again, Venn diagrams. I expect you to be able to do a Venn diagram of a prokaryote and a eukaryote. Okay? Expect a test question to be something like, what are the similarities of a prokaryote? What are some differences between a prokaryote and a eukaryote? Absolutely big distinguishing characteristics that are important to know between the two. All right, tomorrow. We didn't get as far as I wanted, but that's okay. So tomorrow we're going to start using microscopes. So with microscopes, and I know you have used microscopes, but it's been, what, 7th grade?
Yes.
7th grade. So it's been a little while, but nothing's really changed in terms of microscopes. Okay, we got to kind of get some general rules kind of taken care of before we leave today, so we're going to stick around probably right till the end of the day. Okay, so when we use a microscope, compound light microscope, there's 3 different objective lenses, we call them, that rotate around. The smallest one is low power, which happens to be red. Then we go yellow, which is medium, and then blue is the longest. But the longer they are, the higher the magnification. Some general rules of focusing a microscope. Now each microscope is going to be a little different with these knobs, but there's sometimes a big knob and a little knob. Sometimes there's a big knob and then like an inner knob, which is still smaller, but they're kind of like stacked on top of each other. The big knob is called the coarse adjustment, which causes this thing to move up and down quite a bit. Then we use the fine adjustment. So we start with the stage as close as we can. We put the slide under, and then we use the coarse adjustment, and we slowly pull away until we get close, like almost perfect focus. Then we use the fine adjustment knob to get it good. If we want to go to higher power, a lot of times people get this idea like, oh, it's got to be in high power. High power is the best. Not always the case. But if you choose to go to another power, then you rotate, you go red, yellow, blue, low, medium, high. But then from there, you don't touch the coarse knob, you only use the little, because you only got to make minor adjustments, so you only use the fine. If you use the coarse, you're going to make too drastic of changes, you're going to run your lens right into the stage itself or the microscope slide. Then again, high power, we only use the fine adjustment. To make a wet mount. A wet mount is a slide that we go through and we smash between a piece of a slide and a cover slip with some water. So we put a drop of water on our specimen. We take this cover slip, we set the one edge down about a 45-degree angle, and then by just kind of letting it drop on the slide, the water will help hold it. It should then help eliminate some of the air bubbles that we can get. When you change your magnification, I cannot pick this up off the floor. When we change our magnification, no, that didn't work either. The image appears bigger, right? I can't pick it up. I licked it. This is where you probably don't want to touch it.
Oh, wash me.
Okay, so when we change magnification, the image gets bigger, right? Does the object get bigger?
No.
Okay, the object does not get bigger. It just appears bigger, right? We're zooming in. And you do that in a camera, you're like, oh, look, someone's like really far away. I'm gonna zoom in so I can get a better image, I can see it better, but realize it doesn't really make the object bigger. Okay? And what we can do is we can actually go through, and if we know what we call field of view, if we know the field of view, like if I look at my camera, thank you, Mr. Gore. If I look at my camera and I say, hey, from here to here is some distance that I can measure, and then I can say, well, if this from here to here is like, say, 3 feet, and I can fit 1.5 Mr. Davies in that field, how big is Mr. Davies? Well, you take the field of view, 3 feet, and I divide by how many of I can fit through there, 1.5. 3 divided by 1.5, we can then determine the size of Mr. Davies if I know that field of view, which is something we can measure. Okay? And if we change our focus, or I'm sorry, we change our magnification, we change that field of view. But we always start on low power. This is the biggest field of view. Okay? And a nice kind of analogy that goes along with this, some of you may be a deer hunter. And if you go deer hunting with a rifle, you might have a scope on your gun. But if you're hunting for deer and you're looking for deer, do you walk through the woods staring through your scope the whole time saying, gee, I sure hope I see a deer, I sure hope I see a deer, and do you only look through the scope? No, that would be kind of silly, right? You probably would run into a tree, because if I'm looking through a scope, I'm looking at a slice like this big where the deer could be like right here, and I can't see it. So by using low or no magnification, I get a much bigger field of view. I find it, oh, my deer's there, and then I can, like, maybe you have an adjustable scope, or you just pull the scope up, or binoculars, same idea with the binoculars, right? In the baseball game, oh, my friend's out in the center field. Oh, there he is. You don't go like, oh, where's center field? And try to find your friend in center field. It's really, really hard. That's why we always start under low power. Okay? Microscopes tomorrow. We'll look at some pond water on Friday is our plan. Have a great day.